CRISPR and Gene Editing. Imagine a world where we can edit genes as easily as editing a video clip. Welcome to the age of CRISPR and gene editing. For decades, scientists have been fascinated by the idea of manipulating DNA, the very code of life. But how do we edit something so intricate? 1. What is CRISPR and gene editing? CRISPR Cas9 is a revolutionary tool in the field of molecular biology and genetics. It's derived from a natural defense mechanism found in bacteria. Bacteria use CRISPR to fend off viral attacks by remembering viral DNA and then targeting it for destruction. The CRISPR Cas9 system has been adapted for use in other organisms, including humans. It allows scientists to target specific sequences of DNA and either cut them out, replace them, or add new sequences. This precision makes it a powerful tool for genetic research and potential therapeutic applications. 2. How does it work? The Cas9 protein, guided by a custom RNA sequence, searches the DNA for a matching sequence. Once found, it acts like scissors, cutting the DNA at that exact spot. Dr. Jane Smith. Once the DNA is cut, the cell tries to repair it. We can use this repair mechanism to either disable a gene or insert a new one. 3. Germline editing versus somatic editing. There are two primary types of gene editing in humans. Germline editing. This involves editing the genes in eggs, sperm, or very early embryos. Changes made through germline editing are hereditary, meaning they can be passed on to future generations. This is where the most significant ethical concerns arise. Somatic editing. This targets genes in specific tissues or organs of a living person, affecting only the individual and not their offspring. Editing genes in embryos could have several benefits. Eradicating genetic diseases. Conditions like cystic fibrosis, Tay-Sachs disease, or Huntington's disease, which are caused by specific genetic mutations, could be eliminated from the lineage. Reducing risk. For families with histories of certain diseases, like breast cancer linked to BRCA1 mutations, gene editing could reduce or eliminate that risk for their children. 4. Techniques for germline editing. The most notable method for germline editing is the CRISPR Cas9 system. Here's a simplified process. An egg is fertilized by a sperm in a lab, in vitro fertilization. Before the cell starts dividing, the CRISPR Cas9 system is introduced. This system has been programmed to target a specific gene or genes. The Cas9 protein cuts the DNA at the desired location, and the cell's natural repair mechanisms fix the cut. Scientists can leverage this repair process to either turn off a gene or replace it with a different version. 5. Angelina Jolie and the BRCA1 gene. In 2013, Angelina Jolie publicly disclosed her decision to undergo a preventive double mastectomy after discovering she carried a mutation in the BRCA1 gene. This gene mutation significantly increases the risk of breast and ovarian cancer. The potential of CRISPR lies in its ability to possibly correct such mutations at the embryonic stage. This means that, in the future, it might be possible to eliminate certain hereditary diseases from family lines. 6. Stephen Hawking and ALS. Amyotrophic lateral sclerosis, ALS, also known as Lou Gehrig's disease, is a neurodegenerative disorder that affects nerve cells in the brain and spinal cord. Stephen Hawking, one of the most brilliant physicists of our time, lived with this condition. While the exact causes of ALS are not fully understood, certain genetic mutations, like those in the SOD1 gene, have been linked to some forms of the disease. CRISPR offers a potential avenue for targeting and correcting these mutations, which could lead to treatments or even cures for conditions like ALS. 7. Elon Musk and Neuralink. Musk envisions a future where humans merge with AI, enhancing our cognitive abilities. Neuralink is a neurotechnology company co-founded by Elon Musk. Its primary goal is to develop an implantable brain-machine interfaces, BMIs. While Neuralink's work is not directly related to gene editing, the convergence of neurotechnology and genetic engineering could open up unprecedented possibilities. For instance, while Neuralink aims to enhance cognitive abilities through BMIs, combining this with CRISPR could mean editing genes to enhance natural cognitive functions or even to prevent neurodegenerative diseases. 8. The ethical dilemma. The power to edit genes brings forth a myriad of ethical concerns. Playing God. The ability to edit genes, especially in embryos, raises questions about whether humans should have the power to determine genetic destinies. Unintended consequences. Editing genes might have unforeseen side effects, either immediately or down the line in future generations. Socioeconomic disparities. If gene editing becomes a commercial service, it might only be accessible to the wealthy, leading to further societal divides. 9. The future of CRISPR and celebrities. As CRISPR technology becomes more mainstream, it's likely that more public figures will engage with it, either for medical reasons or for enhancements. This could range from therapeutic uses, like preventing hereditary diseases, to more controversial applications, like enhancing physical or cognitive abilities. In summary, CRISPR and gene editing technologies hold immense promise for the future of medicine and human enhancement. However, with these advancements come significant ethical and practical challenges that society must address.